Carbohydrate Catabolism, Cellular Respiration by Zuhair Hassan and Milagros Santos. Cellular respiration. What is cellular respiration? Cellular respiration is a cellular process of oxidizing glucose to obtain energy in the form of ATP. So basically, there are two types of cellular respiration, aerobic and anaerobic respiration. Aerobic means that there is oxygen present to carry out cellular respiration. Anaerobic means that there is no oxygen present. But we will focus only on aerobic in this experiment. The first process of cellular respiration is glycolysis. And glycolysis is known to occur in both aerobic and anaerobic respiration. It's the oldest metabolic pathway used by all cells, and it occurs in the cytoplasm. So basically what happens in glycolysis is a 6-carbon sugar, glucose, is broken down to form 2 molecules of 3-carbon pyruvate. This results into 2 NADHs and 2 net ATP. If oxygen is present, glycolysis then leads us to a second process for aerobic respiration that produces a net total of 36 ATP per molecule of glucose. So this second process is called oxidative respiration, and we can basically break it down into 3 main steps. The first one is the oxidation of pyruvate. Carbon dioxide splits off from pyruvate to produce acetyl-CoA and NADH. And then it goes into the citric acid cycle, which can be the second step of oxidative respiration. It begins with acetyl-CoA, and acetyl-CoA joins oxaloacetic acid to form citric acid, which is oxidized to carbon dioxide. Then this leads to ATP, NADH, and FADH2. Oxaloacetic acid is regenerated for another cycle. This process occurs in the mitochondria of eukaryotes and in the cytoplasm of aerobic prokaryotes. After this process, we have the final step that's called chemoosmotic phosphorylation. In mitochondria, electrons from NADH and FADH2 flow through an electron transport chain from high to low energy states through energy-releasing steps. This establishes an electrochemical proton gradient across the inner membrane of the mitochondria. Then oxygen accepts the electrons to form water, and ATP is synthesized when the H+ proton ions diffuse back across the membrane through the embedded protein. So in our experiment, we derived an experiment to see which set of peas will give off the highest rate of cellular respiration when we looked at germinating peas, ungerminated peas, and germinated boiled peas. And we know that when a seed germinates into a seedling, it needs energy. So to meet this energy requirement, it stores carbohydrates, hence our title, Carbohydrate Catabolism. So these carbohydrates are then catabolized, and the energy released drives the production of ATP. So in our experiment, we were able to measure the rate of the respiration by measuring how much carbon dioxide was produced. So there were 2 parts to our experiment. The first part was our setup and incubation. First, we took the 2 bottles labeled germinated and ungerminated, and we fit a rubber stopper with the attached glass tubes into the respiration bottles. After that, we submerged the rubber tubing into the water-filled test tubes. Then we inserted the rubber stoppers and incubated them for 60 minutes. So, as you can see here, this was our basic setup for the germinated and ungerminated. But then we had the third one, which was our boiled peas. So to do this, we boiled water and we poured in the peas for about 5 to 10 minutes, let it boil for a little while, and then we took them out, drained them, and submerged them in room temperature water to let them cool off. After they cooled down, they were put back into the jar. And then after that, we also let the three of them sit for an hour and become incubated. And then after an hour, we added the phenol red solution, which is shown here, into the 3 test tubes filled with water. And this is our basis for the second part of our experiment, which was measuring the rate of respiration as carbon dioxide was produced. Carbon dioxide gas itself is colorless and odorless, making it difficult to measure. So to measure, we bubbled the carbon dioxide, as shown in the next slide. Here we have a video of bubbling the water. So as mentioned before, carbon dioxide is difficult to measure, and to measure it, we bubbled it with water to produce carbonic acid to help us measure how much carbon dioxide was produced. You can see the procedure here being done in the video. In the 3 test tubes that were previously filled with water, we added the phenol red solution. So that was our pH indicator to measure the amount of carbonic acid. And this in turn measures the amount of carbon dioxide indirectly. So if it turns yellow after the water was put in, this means that it's in an acidic condition. It means that carbon dioxide was produced, and it means that cellular respiration occurred. And as you can see here, we're adding the water. Here's the carbon dioxide bubbling. You can already see in this one had the germinated peas. It turned yellow. This one looks like nothing is happening. This one had the boiled peas. We're putting the stopper here so we don't let any gas come out. And this is our last one. This one was the ungerminated peas. Here we're adding the water to it. Yeah, this one already turned yellow. This one didn't turn anything. And we're waiting for our results for the next one. So, I mean, basically, we did that for all 3 tubes. And before doing this, we had 2 hypotheses. Our first hypothesis was that germinating peas will have the higher rate of cellular respiration. And then when we were preparing for the boiled peas, we decided that the boiled peas will have the highest rate of cellular respiration. So let's go to our next slide and discover our results. So according to our results, our hypothesis was proven wrong. As you can see here, the germinated peas had the highest rate of cellular respiration because it seemed to produce the most yellow in the test tube, indicating a lot of carbon dioxide was in there. The ungerminated peas has a little bit of yellow, indicating that cellular respiration did occur, meaning carbon dioxide was produced, but you can tell it wasn't as much as the germinated peas. The germinated peas that were boiled didn't produce any yellow. And this can be supported by the fact that when you boil peas, the enzymes in the cells will denature, because when cells are put into extreme environments like boiling or even given a higher pH, they will denature, meaning that cellular respiration would not have been able to occur. So therefore, it didn't produce any yellow and no carbon dioxide was produced, meaning no cellular respiration occurred. So therefore, the germinated peas will always have the highest respiration because it needs energy to go into to become a seedling. And that's it for this part of the experiment. In our next prezi, we will explain fermentation. So I hope you enjoyed it.